Okay, let's continue with evolution. This might be the last evolution video lecture. It's the fourth one. We'll see. This is a long chapter. Okay, at the population level, this is where we left off. Biologists focus on the gene pool, which consists of all copies of every type of allele at every locus in all members of the population. But usually, again, just like in genetics, we usually look at like one locus. You got a big letter and a small letter, and there in every member of the population has two alleles, either two big letters, a big letter and a small letter, or two small letters. So what percent of, like if you took all these alleles out of all the individuals of the population and added them all up, what percent are small letter and what percent are the big letter? All right. And it get more complicated in non-Mendelian loci, but now with this, we're just going to look at Mendelian locus that only has two alleles, one completely dominant and one completely recessive. Okay. Okay, imagine a wildflower population with two varieties of blues that have different colors. So there they are, red and, well, it looks like there's some pink there, but let's just go with red and white. But that would be incomplete dominance, and we're not looking at that now. Okay, allele for red flowers, symbolized by big R, is dominant. To allele for white flowers, symbolized by small r. So we're familiar with this. Okay, genotype frequencies. What percent of the gene pool is the big R, and what percent is the small r? And you add those two, it should equal 1, right? So, and it should, this Hardy-Weinberg formula, p squared plus 2p cubed plus q squared equals 1. P is the allele frequency of the dominant allele. Q is the allele frequency of the recessive allele. So p squared is the percent of the population that is homozygous dominant. Big R, big R. Q is the percent of the population that's homozygous recessive. Small r, small r. And 2pq is the percent of the population that is heterozygous. Like we used to calculate the frequencies of genotypes in a gene pool from the frequencies of alleles. Okay. So let's just say the allele frequencies are, big R is 0.8. That means 80% of the alleles in the whole gene pool and the whole population are big R. Small r is 0.2. So 20% of the entire alleles in this entire population at this locus, at the R locus, is smaller. So we do a Punnett square of a heterozygote times a heterozygote. So we got big R right here. That's 0.8 times 0.8, which is 64. So 64% of the population is homozygous dominant. Small r, 0.2 times 0.2 is 0.04. That means 4% of the population is homozygous recessive. And then big R, 0.2 times 0.8 is 0.16, 16%. And there's another 16% over here. And so you add those both together, you get 32%. And so that's where you got these numbers down here. So the percent of the population homozygous dominant, 64%, heterozygous 0.32 or 32%, and homozygous recessive 4%. And they also have to add up to 1, which is 100%. This is often used to find out how many carriers there are, what percent of the people in a population are carriers. usually carriers for like a really serious genetic disease or something. I'm going to show you an example of that in a moment. Well, I'm going to show it to you right now. Public health scientists use the Hardy-Weinberg equation to calculate the percentage of a human population that carries the allele for certain inherited diseases. Okay. PKU, phenylketonuria. Recessive allele prevents the breakdown of the amino acid phenylalanine, and untreated will lead to massive mental retardation. It's treatable, and every one, I think I've mentioned this before in an earlier lecture, every one of us was tested for this within moments of our birth. We tested for it. They take a little blood from our heel and they got tests to see, because it's treatable. As long as you avoid phenylalanine or be careful about how much phenylalanine you have, you can live a normal life. Okay, or you have normal intelligence. Occurs in about one out of every 10,000 babies born in the United States. So what is that? That means that is 0.0001. So that's the percent of the population that has PKU. And so since PKU is recessive, that is the percent of the population that is homozygous recessive. Okay, how do you find out how many people in the United States are actually carriers for it? That's heterozygous, because remember it's recessive, so you can be carried and pass it on to your offspring. You don't have PKU, but you can pass it on. Okay, so 0.0001 is Q squared. So in these problems, you'll always be given Q squared. So, and you can do this math now if you want with the calculator, 0.0001, and take the square root of it. And that'll be 0.01. It's the square root of 0.0001. So that means Q is 1%. So 1% of the entire gene pool at this locus is a small letter. So how many people in the overall population are heterozygous? Well, if Q is 1%, P has to be 99% because it has to equal 1. So 99% of all the alleles at this locus are it's a dominant allele. So in a Hardy-Weinberg equation, what is the percent of the heterozygote is 2pq. So multiply 0.01 times 0.99, double it, and that's what you got. And if you do if you get your calculator now and go ahead and do that, you come up with 0.0198, which rounds off to be about 2%. All right, so 0.02. That means that 2% of the population, of the overall U.S. population, are carriers for PKU. And so that's why on lots of foods, you'll see it'll tell you whether it contains phenylalanine or not. This is for phenylketonuria, people who have this condition and have to be careful about how much phenylalanine they take in. I think I may open up a Zoom session to talk about Hardy-Weinberg problems, because there's probably going to be one on the final exam. Okay. Well, now the other thing you can talk about Hardy-Weinberg is a non-evolving population is in genetic equilibrium, also known as Hardy-Weinberg equilibrium, in which the gene pool remains constant from generation to generation. And the frequencies of P and Q remain constant. So if P is 0.8 and Q is 0.2, then in the next generation, it's still 0.8 and 0.2. And the generation after that, it's still 0.8 and 2. It doesn't change from generation to generations That's Hardy-Weinberg equilibrium. And this means that evolution is not going on at this locus. And the frequencies of homozygous dominance and heterozygotes and homozygous recessive is not changing. But if there is a change, if the allele frequencies do change from generation to generation, that's evolution viewed on its smallest scale. And it's often referred to as microevolution. Generation to generation changes in allele frequencies. So it was 0.8 and 0.2 this generation. Now, for some reason, the frequency of the recessive allele became more common in the next generation and went up from 0.2 to 0.25. Now it was 20%, now it's 25%. And so if that happened, then Q dropped down to 0.75. See what I mean? That would mean microevolution is occurring. The allele frequencies are changing. And this goes on at a lot of gene loci. And this debate and the basis of macroevolution. Now, we're going to get later on, and particularly in the next chapter, we'll talk about what's called macroevolution, which is speciation. And this is the thing. Can these small allele frequencies from generation to generation build up over many, many, many generations, thousands, hundreds of thousands of generations, to lead to the population splitting into two very different types of organisms that no longer interbreed? If this has happened, then microevolution has become macroevolution, speciation. That a population of individuals that is the same species has become two species. Okay, what mechanisms can change a gene pool? Natural selection is the most important because it's the only process that promotes adaptation. But there's several other things that can change a gene pool. Genetic drift is a change in a gene pool of a small population due to chance, some kind of chance thing. So this is a more random thing, and it's called non-adaptive evolution. So here we have back to a population of the red and the white flowers. In this case, the P is 7 and Q is 0.3. So then that's the next generation, it changes. The red, white flowers have somehow been the most successful, and they've spread the small allele into more individuals. So it looks like there's more heterozygotes. There's more white flowers and more heterozygotes. So there's a higher frequency of the small r. So now it's small r is written to 0.5, 50%, and now so P has to be 50%. But then for some reason, well, in this case, only 5% of the 10 plants leave offspring. It just so happens 5 of those just happen to have the small letter. But now only 2 flowers are successful in making babies, and they both happen to be big R. And so that's it. Small has been eliminated from the population. It's at 0. This was just some random chance that somehow pollinations happen to go to these 2 plants that happen to be big R. Well, over here, 5 plants left offspring, and 4 out of the 5 had small r. But here, only 2 plants were successful, and they were big R. All right. The bottleneck effect is an example of genetic drift and results from a drastic reduction in population size. passing through a bottleneck, a severe reduction in population size, decreases the overall genetic variability in a population because at least some alleles are lost from the gene pool just by some chance event. Could be some kind of disaster. This results in a loss of individual variation and hence adaptability. And it's simulated by this. You have a jar of beads, a bottle full of beads. You pour out, you shake out the beads for a few seconds into a glass, and then the surviving population is there. And so the orange allele has been eliminated from the population just because there's some catastrophic event that really reduced the size of the population. And now there's less genetic variability. So cheetahs is a good example. Appear to have experienced at least 2 genetic bottlenecks. If you look at cheetahs, there's very little genetic variability between them. So something happened about 10,000 years ago, something happened that really wiped out a large percentage of cheetahs. And then during the 1800s, farmers hunted the animals to near extinction because they were killing their farm animals. So with so little variability, cheetahs today have a reduced capacity to adapt to environmental challenges and also disease challenges, remember from the red queen hypothesis. So they are living in a precarious state now genetically. And there's a cheetah. The founder effect, likely when a few individuals colonize an isolated habitat, represents genetic drift in a new colony and explains the relatively high frequency of certain inherited disorders in some small human populations. So a small group of this population leaves and goes somewhere, and they happen to have a particular allele, and that allele becomes more common in that population just possibly by random effects. And so this is genetic drift in a small colony, so they become quite different than the population as a whole, and then they get inbred also. And so this is why some certain inherited disorders are much more common in certain ethnic groups, because until recently, you found sexual partners within a small, village group or small ethnic group, and these things. So these conditions were concentrated in those groups. An example is achondroplasia, actually, in Amish people. Amish people usually they're an isolated community. There's not too much contact with the non-Amish. They find the sexual partners that are also Amish, and so this recessive condition is concentrated in that small population. So that's called the founder effect. Gene flow is another source of evolutionary change. It's genetic exchange with another population. It may result in a gain or a loss of alleles, because if there's an allele in this population, if the individuals in this population have this allele and they all leave, then this population doesn't have that allele anymore. This population over here has it. But it tends to reduce genetic differences between populations, because you have an allele over here that can then travel over here. Now both populations have the allele. Before, this population had this strange allele, this population did not. Now you have gene flow, and so you have individuals bring in this allele, and so that's a real genetic difference. This allele over here, not here. You have gene flow of organisms coming into this population that carry this allele. Now both populations have the allele, so now they're not as genetically different as they used to be. So only natural selection consistently leads to adaptive evolution. Evolution that results in a better fit between organisms and the environment. Blue-footed booby reveals examples of adaptations for its home in the Galapagos Islands. Bird's body and bill are streamlined like a torpedo for diving. Its large tail functions as a brake, and female boobies prefer males with the brightest blue feet. This is something called sexual selection that we're going to get into. It's another form of selection. So we've talked about natural selection. We've talked about artificial selection. And now then, in this chapter, we're going to talk about something called sexual selection. That is, that females select the males that they want to breed with. And these males have to have these certain characteristics, or otherwise they're not the females are not attracted to them. So this changes the look of a population. So there's the blue-footed boobies. There's also a different species of boobies called red-footed boobies that have red feet. Okay, relative fitness. Contribution of an individual that an individual makes to the gene pool of the next generation, relative to contributions of other individuals. So survival of the fittest means the organisms that leave the most offspring have the highest fitness, because they affect the gene pool of the next generation more than other individuals that have fewer offspring. So if we graph the color code of a population of mice, we get a bell-shaped curve. If natural selection favors certain fur color phenotypes over others, the population of mice will change over the generations, and 3 general outcomes are possible. Okay, so you have a bell-shaped curve of phenotypes. Directional selection shifts the overall makeup of a population by selecting in favor of one extreme phenotype. Disruptive selection can lead to a balance between 2 or more contrasting phenotypic forms in a population. Disruptive selection so directional selection means one extreme is favored. So the bell-shaped curve moves in one direction. The opposite of that is stabilizing selection, where the extremes are inhibited or lead to lower numbers of individuals. And so the phenotype in the middle is selected. So you end up with a narrow bell-shaped curve and a taller bell-shaped curve. Disruptive selection leads to, means that both extremes are selected. And the middle phenotype is not such. So it ends up with a biconcave shape. And this is the selection that can lead to a speciation event that can break a population into 2 separate populations. And if they stay apart long enough, they won't interbreed with each other anymore, which means a different species. And that's a speciation event. That's macroevolution. Okay, so there is the original population. Fur color. Directional selection means that somehow a darker coat is selected for. And so the bell-shaped curve would just stay the same shape, but it would be moved in one or the other direction, depending upon somehow dark fur is become, maybe the mice moved into a darker area, and now it's harder to see the darker mice in the background. It's easier to see the lighter mice. So the lighter mice get eaten more often than the darker ones. So darker ones have more offspring and pass on those darker genes, and their offspring become even darker. So they move. Over here, stabilizing selection is the medium color, the gray color. For some reason, the light color and the dark color now, they're getting eaten more often or getting disease more often or whatever. Good example of stabilizing selection is human baby weights. The average weight of a newborn baby is around 7.5 pounds, I think. So here's the thing. Really low weight babies have a higher rate of death than the average weight. Also, really heavy babies that are born also have a higher death rate. So they die off more than the middle weight. And so what we have it is a squeezing together of the bell-shaped curve into a steeper, a higher, narrower bell-shaped curve. But then disruptive selection. This is where both extremes are favored. An example of this could be, so you have like an island where you have these plants that make really small seeds, and these plants that make really big seeds. And there's no real plants that make intermediate seeds. So big size, and these birds that eat the seeds, well, the ones with really tiny beaks, they can't crack open the big seeds. So they have to stick to the smaller seeds. The ones with really big beaks can attack the big seeds, and they have much more food in them. And so they don't even bother with the small seeds because it's not even worth their effort. So the ones with the big beaks get really healthy because they eat the large seeds, which the small beaks can't get to. And the ones that eat the small seeds, they get a good benefit from being able to eat all the small seeds. And the big versus the big beaks don't even bother with them. And the ones with intermediate size beaks, they don't know where to go. They're not sure whether they have good trouble with the big seeds, and the small seeds are not really, they're hard to work with that intermediate size beak. It's hard to work with the small seeds, so they become fewer and fewer, and you have this biconcave shape. You have two populations moving apart. And these birds get larger and larger beaks over here, and these birds get smaller and smaller beaks, and eventually they may become so different that they don't intermate, and that's a speciation thing. Okay, sexual selection. Form of natural selection in which individuals with certain traits are more likely than other individuals to obtain mates. And it's usually females do the selection because they make much fewer gametes than males. Eggs are much more valuable than sperm. And so females are much more discriminating as to who they're going to give these eggs to, while males just want to spread sperm all over the place. You know, it's real cheap, it's real plentiful. So, but this has to happen in a population that's sexually dimorphic. Sexual dimorphism is a distinction in appearance between males and females, not directly associated with reproduction or survival. In other words, you get this organism. Can you just by looking at them tell who's female and who's male? That's sexual dimorphism. So are human beings sexually dimorphic? Yeah, they are. Even when they're infants, they're sexually dimorphic. We have different genitalia. So when a baby comes out of mom, you can tell right away, oh, that's a girl or a boy, right? And then they become even more sexually dimorphic after puberty because of secondary sexual characteristics. Females grow large breasts. Males grow facial hair. Their voice deepens. So we're a sexually dimorphic species. But the sexual selection goes on and on. I'm not going to get into that. But some organisms are not, like robins. And they're back now in the spring. And you just by looking at a robin, you cannot tell, just by looking at it, who's male and who's female. They look exactly, they have the same exact coloration. Even if you grab one and look underneath and peer through the feathers, you still can't tell. Robins have no external genitalia. They have a cloaca with 3 openings in there: an anus, a urethral opening, and a genital opening. That there's either eggs in there or sperm is going to come out of it, and you can't tell which are looking at it. So they are not sexually dimorphic. Sometimes sexual dimorphism can be really extreme, you know. Sometimes it's just a little bit. And actually, the more sexually dimorphic there are, and the more strange ways that the male has, the more sexual selection there is. Okay. Okay. In some species, secondary sex structures may be used to compete with members of the same sex, usually males, for mates. You know that these wide-eyed flies, they have eyes at the end of these long stalks, and two males compare eyes, and the female's waiting nearby, and the one that has the widest eyes gets the female. Females like really wide eyes, and that's why they have wide eyes. They involve physical combat, like mountain sheep. They bash their head together, and the one that comes out wins the battle, gets the female. That's also like physical combat. So a lot of times also male spiders will fight each other on the edge of a female web, and the winner gets to be the mate with a female. But oftentimes they're ritualized, like the comparing of the eye widths. They don't really have combat, but they have ritualized combat, and one of them wins. Okay. In a more common type of sexual selection, so that's sexual selection also, but it's combat or comparison between males. More common type is that individuals of one sex, usually females, are choosy in selecting their mates. And they want mates that want males that look in a particular way, right? And so if they don't look in a particular way, they're not going to be mating with the female. So there's sexual dimorphism in a finch species. The more colorful one is usually the male, or competing for mates within these antelope here. To talk a little more about sexual selection, because okay, you can see the combat. That means the strongest male gets to mate with the female. Right? But what about these other things like peacocks and peahens is a really big example of that. The male peacocks have this gigantic tail, and they display it to the female. And if she really likes it, then he gets to mate with it. Why would females want a male with a big tail like that? Or even common in our own environment, cardinals. Male cardinal is brilliant red. Female cardinal is just like reddish, much duller red. But and only the brightest, most brilliant males get to mate with the females. But why do females want the really bright color? Is just because they're more attractive? Oh, I like red. Or does that signify something? It indicates that the male might have some good genes, because for two reasons. One thing, usually in sexual selection, the male has a deficit. The male is given a deficit. And if he can survive with that deficit, then maybe he's got some smarts, or maybe he's adaptive, or maybe he's got some good genes. Because that cardinal is sitting out there, usually want to be camouflaged against predators. Well, the brilliant red cardinal is not camouflaged. He sits like a sore thumb. You can see him a mile away. So if he's that obvious to predators and he's still alive, maybe he's got some smarts, maybe he knows how to get around, and maybe he's got good genes. Second thing is, it's expensive to make those red colors. That takes a lot of energy to do that, which means food. If you're not well fed, you're not making that really bright color. So maybe you're good at getting food. And so if I mate with you, then my male offspring will be good at getting food, and they're going to be able to get mates on their own and pass on my genes. And also immune systems. If you're sickly, you're not going to be making that nice red color. So if you got that nice red color, you probably got a pretty good immune system. And that means my male offspring, if I mate with you, will also have a good immune system, probably. My favorite is fiddler crabs. Female fiddler crabs have two small claws that they feed themselves with. Males have one small claw that they can feed themselves with. The other is gigantic. This huge, useless claw. It's just hanging there. And it's tough to get around with it. But females, so this is a deficit. He can't feed himself with this, and it's hard to get around with. But females want one big claw. And so it's really funny to see this when a female comes along and all the males in their burrows, the males are waving this huge claw, trying to impress the females. Say, hey, look at this. In this case, size does matter. And if the female is sufficiently impressed, the female will go into the burrow and they'll have sex, which means that the male passes this spermatophore into the female's genital opening, and the female leaves, and then the male's out there waving that thing again to try to find another female. And mostly in sexual selection, males are strictly sperm donors. That's all they do, sperm donors. They have nothing to do with raising the offspring. All right. And so there's a relationship actually because how extreme this sexual selection is and male parental care. Usually in a really strong sexual selection, there's no male parental care. But in species with no sexual dimorphism, and therefore no sexual selection, males help with the offspring. Male robins help with the nest. They bring food, they help clean the nest. The only thing they don't do is incubate the offspring or incubate the eggs. The female does that. Well, back to the peacock. It was a big tail. Is that a deficit? Well, yeah. It's such a huge thing lying back here. Male peacocks have a real tough time flying with that huge tail trailing behind them. And so this is a deficit for the peacock. So this is how sexual selection is thought to work. Okay. Evolution connection, threat of antibiotic resistance. Antibiotics, drugs that kill infectious organisms. I think we talked about this before, first discovered in the 1940s. But by 1952, some bacteria had genetic traits that enabled them to resist the killing power of drugs. So as I said before, if you get an infection now and you want to go to the doctor to get an antibiotic, you don't get penicillin anymore. It just doesn't work. Same thing that, the same way that pesticides select for resistant insects, antibiotics select for resistant bacteria. So our enthusiasm for these things has encouraged the evolution of antibiotic-resistant bacteria. Like, this is crazy. Livestock producers add antibiotics to animal feed. I mean, that is so stupid. And so this, I don't understand why the government allows this. That they must be passing money out to congressmen and senators because this is really a bad idea. I mean, we have to go to a doctor to get antibiotics, and we get them in little pills. They go to the feed store and buy, kilogram quantities of antibiotics to feed the livestock. It's crazy. Doctors may overprescribe antibiotics. They don't do that much anymore, but they used to. A parent will bring their kid to a doctor because he's got a bad cold, he's got the flu or something, and they give him antibiotics. Useless. Cold and flu, it's viral. It's not bacteria. Antibiotics only work against bacteria, but I guess it makes the parent feel good or something. And maybe have a placebo effect, maybe. But you overuse, the more you use antibiotic, the more you're going to breed antibiotic-resistant bacteria. Also, people may not take the full course of antibiotics, allowing mutant bacteria that may be killed more slowly by a drug to survive and multiply. So you should use if you go to the doctor and get antibiotics, you can start feeling better, use the whole supply. Okay, so this is particularly strong in hospitals where antibiotic use is extensive. So a formidable superdrug known as MRSA can cause potentially fatal systemic infections, often begins in community settings such as athletic facilities, schools, and military barracks. And it's really resistant to antibiotics. In fact, I think it's an acronym for methicillin-resistant Staphylococcus aureus. and it's a very common bacterium, and this is one that's resistant to a lot of antibiotics. So it's really hard to get rid of if you get it. So it gives you indications in locker rooms and everything in settings to be careful. Well, we're careful now because of coronavirus. You should be careful for any place where this bug might be found, particularly in hospitals. That's why I'm a little hesitant to go into hospitals. My sister was in the hospital for quite a while, and she got MRSA. She came down positive for MRSA, and when we visited her, we had to wear all these gowns and masks and gloves and everything. So hospitals are a really good place to get MRSA. Okay, that's it. So we're just back to figures now of earlier stuff that we looked at. So overproduction of offspring, individual variation, natural selection, unequal reproductive success. Hardy-Weinberg, different forms of natural selection, directional selection, disruptive selection, stabilizing selection. And that's it. So we're done. So it took 4 so this is going to be 106 evolve 4. And we're done with chapter 13. And so there's just one more to do. Next video chapters will be on chapter 14, which deals a little bit more with evolution, more with macroevolution. This chapter was mostly dealing with microevolution, natural selection. The next one we'll look at more at macroevolution, speciation, and speciation isolating mechanisms, and the diversity of life on Earth. Okay, that'll be chapter 14. So I'll see you later.